ฮัลโหลอ่ะต้องใช้2 อันเลยใช่ไหมโอเค Okay. Is this good? It's good, right? Where is the camera? That one, right? Can you hear me okay? Good. Okay. So, thank you so much, everyone, for coming. Actually, there should be 2 more professor coming, อาจารย์ Professor Thana and Professor Kon Kanya that uh, the speaker and I met yesterday. So, anyway, welcome all of you. Actually, there are like 10 students, 10 participants in this workshop, but most of them cannot join the on-site. So maybe just 3 of you being the lucky one to join on site. Anyway, for those who come on site, we will leave for lunch at noon. Then we will have lunch with the 2 professors here. So just a little bit of introduction. The 2 speakers that come today, I could say that they are the leading scientists in using AI together with computational chemistry. So, in terms of computational chemistry, the conventional way, we use the method called density functional theory. You may heard that you may heard that many times. Density functional theory, it is already a standard in quantum chemistry and quantum physics. If you want to calculate like energy and interaction, blah 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 blah, you use DFT, right? But DFT is very slow. um, for example, in my own research, when I want to run DFT, I have to use supercomputer uh, in National Thai Association, something. Yeah, sorry, I can't remember. NASDAQ, and I have to pay a lot of money, like 50,000 baht for the research unit, for the compute unit, and it run for many days or sometimes weeks. We hope that when we use AI, it will shorten that time to be much faster, but at the same level of accuracy as DFT. So now we have another professor coming. So good morning, อาจารย์กรกัญญา for coming in. Okay. So without further ado, okay, we have one more coming in. อาจารย์ธนา thank you so much for coming in. Okay, without further ado, I would like to welcome our speaker, Dr. Kenny Kaiser Custodio, right? Am I pronouncing your name correctly? Yeah, okay. He's actually from the Philippines, and right now he is a researcher at Academia Sinica in Taiwan. Academia Sinica is somewhat like uh, Sawater in Thai. I'm not sure how to pronounce, how to call Sawater in English. NSTDA, National Science and Technology Development Agency, something like that. Okay, I have been speaking too much already, so please welcome Dr. Kenny to the floor. And you can put this on your shirt.
Yep.
Okay, and here.
Okay, all right. Okay, so thank you, everyone. Actually, this is not like, I mean, pretty much everyone knows it's not like the first day of this workshop. It's actually the last day, and we're kind of like wrapping up the whole thing. So we'll not be running like all 3 hours. Actually, I was just given like an hour and a half. And the first part would be just wrapping up everything that we have discussed so far for the last 4 weeks. And the second part would be discussing more on what I have personally have participated in our research in Taiwan. Yeah. So how do I? This one, right? Okay. So just a quick review of what we have kind of like discussed a bit. I know the first day we had like a 3 hour discussion on like just the introduction, the concepts behind everything that we have done so far. But essentially, when we try to explore the conformational space of let's say any system, or in this case complex systems, there are several ways we can do that computationally, right? So this is quite what do you call this? Very simplified, but essentially, if you're gonna ask me, I can just group the two methods in calculating or in exploring the conformational space. So first is through the use of the classical force fields. So classical force fields are usually used for larger systems, right? And for longer time scales. So this is quite important when we try to uh, predict the bulk properties of materials. They're very uh, popular in that aspect, as well as liquid systems. Okay. And then we have the more expensive one, which is the ab initio method. So one type of ab initio method is DFT, density functional theory, but it's not just limited to DFT. You can also have like couple clustered calculations as well. So, but essentially, ab initio means first principles. That means it also includes, or it tries to explicitly describe the electron behavior. Because for classical force fields, electron behavior is pretty much parameterized, and that means that the relative accuracy of classical force fields with respect to like the first principles calculations is much uh, uh, the error is much wider, right? But since we're but for both of them, they have their own niche, right? So bigger systems, we want to use classical force fields. For more accurate description of systems, but usually this means that you have to limit the size to really, really small. And so it can go into reaction mechanisms, uh, very specific conformations, those things. And in some cases, they are able to predict spectroscopic properties, such as like through NMR or UV-Vis, IR, and so on. Yeah. Okay. So where does NNP fit in? Now, if we look at force field, as I've mentioned, this is quite cheap. It's very fast because it's parameterized, but at the cost of accuracy. So it's relatively low accuracy. On the other extreme end, you have ab initio, which uses quantum mechanics, and that means that it's quite expensive, but then you get high accuracy, right? Now, what we want to bridge here with NNP is this big gap here. And so if we look at this uh, uh, kind of like this is a general rough uh, estimate of the accuracy versus cost. You see the force field kind of sits here, and then you have semi-empirical force field similar to traditional force field. And then at the other extreme end, you have the ab initio methods. What we want here is that we have a lower cost for ab initio, but with the lower cost with respect to ab initio while having a very similar accuracy. That's why it sits right here. Okay, so in essence, we want to get the best of both worlds, from the cheap and fast force field usage and the accuracy of ab initio. Of course, it sits right in the middle, so it's not exactly as fast as force field, but not as accurate as ab initio. But as you can see here, we try to get NNP as close as possible to the ab initio accuracy. Okay, now this one is very complicated, and what I wanted to show here is that in our current research in our group, we're using this particular architecture, the Chinet architecture. And I know the title of the workshop is more on like Chinet, but what I wanted to highlight here is that this workflow, we hope, is not just applicable to Shinet, right? Because what we want is transferability of skills, of research skills. So if we want that transferability, we should not limit ourselves to Shinet, right? And I hope for the participants, what you get from this workshop is not just on the usage of Shinet architecture as it is, but the way we attack the problem, how we prepare these, for example, the data sets, how it is used to perform the structure search, for example. Yeah. But anyway, I just show here, this is a figure from the actual research paper which published the Shinet architecture. And you can see here there are several layers, and this is essentially the workflow inside the architecture, how it predicts the energy and the forces. And also I wanted to highlight here that this particular architecture uses this graph neural network. So this is a bit more technical. There's a lot of things that needed to be discussed if you want to know more about graph neural network. But I think that is reserved for another time. Yeah. Okay. This one I have shown already. So what we want to do here is to use this particular architecture in order to create a model, and this model should be trained on a target ab initio method or a DFT method to a certain degree of accuracy. And later on, we can see how accurate our models can be. And this one here, we want to use that to accelerate structure search. So how exactly are we doing that? Yeah. So let's say, for example, this is the black line here is the potential energy surface of the at DFT level. If we train our neural network potential model on that DFT, then we can sort of like have a similar shape of the potential energy surface, right? To some accuracy, right? So that means that we can do this more simplified approach. So we can just start with the guess structure. These are initial guess structures. Essentially, this is the sampling part, right? For those who are doing computational chemistry, this is where we kind of like try and prepare these different structures that we optimize at the DFT level. Now, the more complex the structure is, the more guesses that you have to make, right? And so for complex structures, you would expect you have a lot of this, right? But in traditional approach, we usually make compromises. Like maybe we can remove these types of structures that are we think are quite far from the expected low energy minima, for example. So we make those decisions. But with NNP, we don't need to make those compromises. We can start with a large pool of structures. We perform this called geometry optimization. So we obtain these local minima. So local minima are those that sits at the bottom of each well. And then when we get that ensemble of NNP minima, then we can just select a few of them, which we call them like candidate structures. And those are the ones that we calculate with DFT. So what's the advantage here? The advantage here is that you're not doing direct DFT optimization from a relatively high energy conformer, because that means that you will take more steps, right? So each step of the optimization costs some amount of a computational resource. But if you start at the level of NNP minima, then the number of steps can be decreased a lot, right? And so even though we're still using DFT at the end, we are not using many steps, right? And of course, the other thing is that we're not losing more structures because we don't have to make these compromises wherein we want to decrease the number of initial guess structures. Okay, so as I've mentioned, complex systems are what NNP are really good for. Well, at least in our research, but of course, it's not limited to that. And in this case, in our group, we're actually looking at glycans or carbohydrates and then peptides. Right? As you can see, you have a lot of single bonds. And single bonds mean that they can be rotated. And so sampling will take much more. Right? Yeah. So in this, actually the one that I'll be presenting later, I'll be focusing on glycans. This is what I have been working on. But we have group members. Actually, our PhD student who's recently just got his degree worked on peptides, and he worked for a length of peptide as long as 6 peptide length, and that's actually a big leap because we're talking about DFT accuracy, right? And that was only possible, or at least reasonable, only when using NNP in our case. Okay, so we're actually quite nearing this, ending the first part, but I just wanted to show you again the workflow that we have been doing. We have been adapting in our research. So first, we have the structure. The first part would be well, this is not actually part of it, but anyway. So we start with the sampling here, and then some of the samples we take a portion of it, and then we generate the training set. Right now, there's a lot of things that need to be considered here, such as the diversity of the system, um, choosing the right geometric features, and then when after preparing the training set, then we train our model. We get our model here, and then this model now will be used for geometry optimization of the samples that we got from this part here, and then we will accumulate all those local minima at the NNP. So you can think of NNP as just similar to a DFT, right? NNP is just DFT that is not really DFT, if you get what I mean. Okay, and then we get these some of the structures here, not all of them. Just choose those low energy structures, and then we optimize them at DFT, right? And so at the end of the day, we still have structures or low energy structures that are at ab initio level. Still DFT level, they are not NNP level. And so we can still say that they are first principles. Okay? And yeah, so in closing, for us, we look at NNP as being a powerful tool, and we treat it as a tool to speed up the rather computationally expensive, quite time-consuming structure search, and that for us, it really shines when you have a very complex structure, and when you need a high accurate description of the potential energy surface. So it's not always that we have to use NNP. NNP is still more expensive than semi-empirical calculations and traditional force fields, but since I'm assuming we have experience with chemistry or physics. Sometimes we really need to zoom in on those specific interactions, specific conformations, and so we sometimes demand higher accuracy in the form of DFT, for example. And so I think NNP can really help support that goal. So as Professor Non had mentioned earlier, it takes a lot of time to calculate with DFT, right? Sometimes we really cut corners. And so hopefully with NNP, we don't really need to have to do that. And with the age of AI today, it would be a shame not to really take advantage of AI tools such as NNP. Okay, so I think that's it. So do you guys have questions so far? Yes. Do you?
Okay, so yeah, so uh, let me talk about ask you about the speed, right? So so I have seen the workflow and some of your examples that you try to train the NNP model from glycans or peptides.
Yes.
But then okay, that we can train the model using a lot of conformational samplings of small molecules and short peptides. Okay,
Right.
and then I'm thinking about how to implement the force field or the potential with let's say the larger peptide, the longer peptides, or glycans with many units.
Right. Right.
Or sometimes it could be the idea is that okay, if we train one model that can describe the properties of glycan, and another model that can describe the peptide. But if the system that we want to simulate using potential would contain both glycan and peptide, like glycopeptides, can we do that, like in principle?
Right. Oh, okay. Yeah, so actually, that's a very good question. And luckily, at least for me, I've already seen this. Also, one of our members in our group is actually working on glycopeptide involving leptins and some, I think it's mannose, alpha mannose system. So we are actually already doing that. And so this is where the challenge comes in, right? Because at least the way we use the Shinet architecture, we limit the usage of the NNP model to the target system. Now, the compromise there is that means the model will only be useful for that particular system, but it has a relatively high accuracy. So for now, what the routine is that, oh, you need to work on the system, then you have to prepare the data set to train on this target system. Now, if we have, let's say, NNP that works on the peptide, a particular peptide, and then another NNP for the glycan, what we usually do is we do the sampling from the database that was generated from these NNP minima, and then we perform this specific sampling. Well, in general we call it pattern transfer, wherein we try to like assemble them, not really docking, but like just put them very close to each other, and then there are several ways we can go about it. Either we do some sort of like another type of sampling. Actually, I'm not sure if you're familiar. There is CREST sampling, wherein it uses metadynamics approach in order to sample. There's of course the more manual way of doing it, just using a script to put them together. But essentially, at the end of the day, we still need those single point DFT data that is obtained from the actual interaction of the glycopeptides. So that's actually one of the limits that we are currently trying to break through right now. But yeah, that's actually a very good question, because this is a lot closer to experimental data. And then, of course, we always want to do these collaborations with them. So we have to be more reasonable with how we design our systems. But yes, I hope that answered the question. Right.
So just like also depends on sampling strategy.
Yes. So that's actually a good point as well. The majority of decision making that goes into this is not the training. It's actually the sampling and and of course, by sampling, by extension, we also need to think about how we prepare the data set. Which data points are important? So for us, for example, if we want to cover more of the potential energy surface at the low energy region, then we make a cutoff, for example, of the relative energy of the data that we want to put into the training set. So there's a lot of decision making that goes into it. For now, I cannot say definitely this is the only way to do it. But of course, this is research. We always try new things. We always try to push the limit. And so, yeah, this is something that we have to really think about when we train. Yes. Thank you. So you guys have more questions? Yeah. So now I would want to go to share actual research that we have done so far. Yeah, so actually this is an edited version of what I have presented back in Taiwan, but essentially, actually some parts of this one is something that I've already talked about, but let's see. Okay, so by introduction, since I've mentioned that I have been working on glycans, so I'll be focusing more on glycans. Now, the thing with glycans is that they are very complex, very flexible structure. And in terms of cell recognition, these different conformations can have different impact, right? And within these glycans, you can have different isomers, right? So if you know your organic chemistry, you'd know that the form of, let's say, the pyranose, for example, can have different isomerization depending on whether the hydroxyl is up or down. So, for example, here, right? So, they look kind of the same, but if you look closely, the relative positions of the hydroxyl and the hydrogen atoms, for example, that defines the isomer, right? And different isomers would have different interactions, for example, in a glycopeptide system. And that's important for like these binding interactions, for example, or as a receptor complex. So this is something that's quite important. Now, analytically, this is hard to determine only by using mass, right? So if you run mass spectroscopy, for example, mass spectrometry, you would see that these different isomers would have the same mass. So mass is out of the picture if we want to distinguish them. And so that means that we should look into other ways to distinguish them. And one way is to look at the infrared spectroscopy. So infrared spectroscopy looks into the vibrational signals based on like the bond stretching profiles and bond bending in some cases for these systems. And and what we want to show here is that IR spectroscopy can be used to distinguish them, right? And the thing with IR is that these bond stretching are highly sensitive to the overall 3D conformation. So if we are using this as an analytical tool, we want to know what the structure looks like, right? And usually in an actual system, these structures are the low energy structures, right? And so exploring the conformational space is quite important. Okay, now the issue here is that first, we are we admit that the experiments can be very limited, right? And not only limited to the availability of, let's say, an instrument, but also the availability of the actual sugars. So usually you will be limited to what's already available commercially. And so it would be hard to, let's say, test on a sugar that needs to be synthesized first, right? The other thing is that if we really want to get like a really high resolution spectra, usually we go to cryogenic conditions, really cold conditions, so that we limit the number of conformers that we observe when we measure these IR spectra. So for example, here, this is not from our work, but from this work, where they used IR multiphoton dissociation spectroscopy. And you see like there's like wide bands, right? And so in this case, it might be a little bit challenging to really distinguish them from each other. What I'm saying is that if we don't know these labels, for example, if we only put them in the IR, for example, without knowing their identity, it's hard to say that this is, let's say, the alpha conformer of this sugar and this is the beta, because they kind of like look very similar. So in some cases, like lowering the temperature can give us better resolution, right? Now, even after saying that, there is actually one way that is more accessible, at least for us computational scientists, wherein we can use molecular modeling to try and predict what structures correspond to these systems, for example. And the idea here is that if we can build a collection of these structures, or build a structured database of these low energy conformers, then we can make these predictions that, oh, this peak here corresponds to this bond stretching of this particular conformer, or this low energy conformer. Now the problem is glycans, like as I mentioned earlier, they're quite flexible, and that means that would require more effort in sampling. Okay, so in one of the earlier papers of this French group here, led by Dr. Isabelle Compagnon, they have done this. They have done the experimental and computational simulation of this particular system via IRMPD and ab initio calculation. So they look into the protonated and acetylated hexosamine. So you don't need to worry too much about the actual structure, but know that this is a pyranose. This is a 6-membered ring sugar. And this is their computational approach. So first, they ran molecular dynamic simulations using PM7, which is a semi-empirical force field, at a very high temperature. So the idea here is that if you run a system at very high temperature, then it can access a lot of conformations, right? Now, what they did is from here, they took out only 1,000. Now, this number here, we think this is quite, I mean, we have a question here like, is it enough, right? Because the thing here is that you start with 1,000 here, you re-optimize the structure at the same semi-empirical force field. So you get a minima here. And now your 1,000 structures are reduced to 100 plus structures. Now take note that this is semi-empirical. And with relative to DFT, the error can be as high as 30 kilojoules per mole, right? You have an error of 30 kilojoules per mole, and you're only getting 100 plus structures there, right? And then from this 100 plus structures, at DFT level, but at so within the DFT functionals, each functional will have a different cost from each other. They don't have the same cost. So this one here is relatively cheaper DFT functional. And then whatever they got from here, they further optimize this as the more expensive functional and basis set here. So the idea here is that they had to go several layers of reoptimization just to make sure that at the last step, because this is the most expensive one, they only choose a few of them. So you do realize that in this manner, it is quite possible to lose a lot of structures, maybe the important structures at around this part of the scheme. But what can you do? They're not using anything else but this traditional approach. And so they had to cut some corners. Now, what's the consequence of these cutting of corners? So for example, here, they have this experimental spectra, right? And below are the predicted calculated structures. Now, the way we usually do this is just that we match the peaks from the calculation with what is observed, so we can attribute which particular conformer is actually measured in the experiment. But in this particular example, this system here, right, they were not able to really account for this small peak here. So what's the speculation here? Now we speculate that perhaps this is actually a missed low energy conformer, and that could have been due to this scheme, right? It is not wrong. What they did is not wrong, but we think that maybe we can improve this method that they have by using neural network potentials. Okay. So yeah, so actually I've already shown this. Essentially, what we want is the NNP to be able to mimic the potential energy surface of an ab initio method, a highly accurate method, for example, DFT, by predicting both energy and atomic forces. So it's not just energy, but also the atomic forces. So in my calculation, like at least the efficiency that we get from here is at least 100 times compared to the traditional approach. Okay, so at least 100 times. So, but if we increase the complexity of the system, this can go higher. Okay, so if you, let's say you have a disaccharide instead of a monosaccharide, then you expect that this complexity, efficiency here would go as high as 300%. Sorry, 300 times, not percent. Okay, so yeah, so this would be an example of how the neural network is calculating, but we won't be going to the details. This is very technical, but yeah, this is an example of a neural network. And of course, it's called neural network because it is inspired by the neurons inside the body, from humans. Okay, so the way we do the NLP training, essentially the data looks like this. So the training data needs to have the atomic coordinates and the corresponding electronic energy and atomic forces. So in one of the videos that we have pre-recorded, you can see that inside I think we're using the XYZ file structure. So it has the energy and then the coordinates and the atomic force vectors. Yeah. So we need all those three because we are training our model on all of those input. Now, in our at least in the ones that I have worked with, we have used two types of approaches in generating the data. So first is from scratch. This only happens at the very beginning, right? Because if we don't have any starting data that's suitable for training, for example, for generating the database, the data set, the training data set, then we'll have to do from scratch approach. But in our current state, we can actually do the pattern transfer. Pattern transfer is where we have a starting relatively decent pre-model of the NNP and a starting structured database. So all we have to do is perform some sort of sampling, pattern transfer sampling in order to create these data. Now, just to give you a short idea, so in these two works that I'll be showing you, I actually used this particular version of Shinet. So in the tutorials, the videos that you have seen, we're using Shinet Pack 0.3.1 version. Okay, but actually currently they have a more advanced one, which is the Shinet 2.1.1. So you can you can also explore, but the thing is that they're using a different structure in their architecture. So you might need to have a closer look on how they exactly implement it. But as I've mentioned, the workflow is still the same. Okay. All right. So when we don't have any starting database, we don't have any good, decent pre-model, we do this sort of like a from scratch approach. And so, for example, here we can do some random sampling of the structures. When we say random sampling, we just randomly rotate the single bonds present in the structure. So you can do rotation of bonds, and then you can also do some improper torsional angle rotations in order to explore the ring puckering, for example, of the cyclic sugar. Next, we can perform some clustering of the structures. So you generate a lot of these structures, right? And they will have different ring puckering, different if it's protonated structure, then the protonation can go at different atoms, for example, or even looking into the intramolecular hydrogen bonding. So at least these are the things that we considered. So we can look at hydrogen bonding, protonation sites if it's protonated system, or even ion coordination if we're using ions in our system. So the idea here is that we want to maximize structural diversity. Okay, so when we generate our data set, it should have structures that are very distinct from each other and as much as possible covering most of the potential energy surface. Okay, and then lastly, we just do this batching of structures. So the idea is that we're just taking a small portion of the samples. Of course, it doesn't make sense if we train the model on all our samples, right? Because that means we just might as well just run DFT, right? But we just take a small portion, which we believe is representative of the entire conformational space that we want to cover. And that's gonna be our data. So let's say we take one batch of those structures. The idea here is that since we don't have any NNP model, we have to perform some DFT optimization. Now, the thing with DFT is that if you wait for it to converge to the most stable conformation, it can take quite some time, right? So for example, here, if we look, this is, for example, energy. I forgot to put the y-axis. So this is energy here, and this is the number of steps. You can see that as you go past, let's say, 10 steps, right, or maybe 20 steps, depending on the structure, the changes in the overall conformation is very limited, right? And what does that mean? That means that this collection of structures here are very, very much similar to each other. So we don't really want to spend too much time waiting for them to converge, just to prepare our data set. And so our strategy here is that we can limit the number of steps that our initial guess structures would take to undergo DFT. And so here we selected 10 steps. This is based on our previous testing of the trajectories, the DFT trajectories, and we find that just collecting the first 10 steps would be decent, would be enough in order to create our initial data set. Okay, and then of course from the trajectory, the 10-step trajectory, we perform some sort of screening. So those that have done this part of the workshop, you know what structure screening means. So this undergoes structure checking and duplicate removal. And then we accumulate our training data, right? And then from this training data, we can now start training our NNP. So we call this, for example, just NNP1. This is the first NNP that we have generated. Okay, next. Right, now, when we talk about pattern transfer, so this is when you already have a decent model, right? You have a decent pre-model, and then you already have a like a source or reference structure. So for example, we want to study galactose structure, right? And then we have already the glucose database. Now, if you know the difference between glucose and galactose, they only have a difference in the position at the carbon 4, like the hydroxyl and the hydrogen. So it might be easier to just start with glucose, but we perform some sort of like flipping, right? So that's what we call the flip and rotate, right? So what's the idea there? We assume that this flipping has a very, very marginal or relatively small perturbative effect, right? And so the optimization would not take too long or would not take too many steps to re-optimize itself to get to its lower energy or lower, a more stable conformation, right? So now we're not just limited to flip and rotate. So for example, we want to methylate the system. So let's say you have glucose system and you want to methylate at carbon 1 to stop it from or to inhibit its mutant rotation, for example, then we can do some sort of like attach and rotate. In some cases, you want to you start with a neutral system. You want to put in an ion in there, then you can just randomly place the ion around it. So what I'm saying is that in our case, we have prepared like tools in order to do these pattern transfer approach. Okay, so the same thing. Instead of doing random sampling, as I've shown in the previous slide, we can just do this sort of pattern transfer sampling here. And then we still do the same method of cluster and batching. Okay, but the difference now is that we don't need to do direct DFT because we already have an NNP model, right? So the idea here is that we always want to use what's already available. So what we do is that we take the NNP model, the pre-model that's already there, we perform optimization, right? So this is where the DFT optimization was earlier. Now we replace it with NNP, and then we collect the NNP trajectory, right? Now, the thing is that the training set should still be DFT level, right? So we cannot just take the NNP trajectory snapshots and then use that as the training set. We still need to calculate the DFT level energy and forces at those particular snapshots or conformers. That's why we have another step here of calculating the single point DFT of the NNP trajectory snapshots. So at the end of the day, our data set still contains DFT energy and forces data. Okay, and then with that, we can now train our NNP. So these are the two approaches that we have implemented depending on the initial conditions that we have. Okay, so of course, if we do training, we also need to test them out, right? And so what we do is that we prepare these different batches, and each batch maximizes the structural diversity. So for example, if we don't have any NNP or we just have a pre-model NNP, so the training set, that would be from the batch 1. And then we will also actually run the same process to create batch 2. So essentially, we have two sets of data. One would be used as the training, and the other one would be for testing. So we will test the model that we have trained on this batch here. Now, let's say, for example, from the testing, we see that, oh, there are a lot of these outliers, right? So that means the model is not good enough yet. So what do? What we do is we take those test sets. So in practice, we take the outliers and then we add that to the training set. So this is what we call active learning approach, right? So we continually improve the NNP by adding in those data that were not predicted well by the model until you get to a point wherein the overall, we call this MAE, this is mean absolute error. The mean absolute error reaches a particular threshold that you set. For us, we usually set this at 2 kilojoule per mole or 3 kilojoules per mole. It depends on the size of the system or complexity of the system. So as you can see here, you can continue to do this process of like refining the NNP until you get what you get an NNP with your target error or target accuracy. Okay, right. Oh, sorry. Questions so far? I think I've been rambling on for quite some time. But any questions? Okay, maybe your questions you may think about them later, but yeah. Okay, so for example here, this is how we kind of test out the accuracy. Sorry, you cannot really look very closely, but here, let's say we start with the pre-model, right? And our target system is this alpha galactose N-acetylated that's protonated. And the pre-model, which was trained on glucose, gives us an error of around 3.6 kilojoules per mole for energy and 3.5 kilojoules per mole per angstrom for the atomic forces. When we train this NNP by adding in more of the data from galactose, we have now improved the accuracy to 1.4 for energy and 1.8 for atomic forces. Now, I'm saying these numbers, but what do they exactly mean, right? So remember earlier, maybe I can go back a bit. It's not here, sorry. Yeah, remember this overlay of the DFT and NNP, right? So MAE here, so energy, let's say EMAE, so MAE of the energy. That means that's on average the distance, the relative distance of the measured energy predicted by NNP compared to the actual DFT level, right? So the smaller this MAE is, the closer the NNP is describing or mimicking the DFT surface. And remember, this is at least 100 times faster. So you can see how good NNP can be. Okay, so now going to the meat of the whole thing. This is the actual research that we have done so far. So this is the first paper that I have published under Professor Guo with NNP. And here we have looked into the conformational space of protonated and acetyl hexosamines. Now, for sugars, usually it's easy to study the more common ones. You have the glucose, galactose, mannose. They are more naturally occurring, I might say, right? But if we look closely at these structures, there are actually just 3 out of the 8 possible conformers, right? So this cube represents the different isomerisms that these systems can have access to. So you have allose, gulose, idose, altros, and talos, for example. And we're not even talking about the anomeric forms. You can have the alpha and beta conformations as well. So in total, there could be 16 potential isomerisms. So actually, no, 32 if we also consider the L and the D at carbon 6, right? But anyway, we'll just focus on the 16. So all of them are D forms of these sugars. Now, the table that I'm showing here is actually the process or the results of our training. And there's too many to look at here, but I think the most important one to really pay attention to is this last NNP here, because what I have done here is that I have trained a single model which covers all 16 systems. And this is the relative accuracy. So all of them are less than less than 1.76 kilojoules per mole accuracy for the energy, and for the forces, it's less than 1.88. Right, so very, very accurate, relatively, at least in the low energy region. Okay. So now I think I've been asked many times, right, regarding how do you know it actually works, right? So what we did, as I've explained earlier, when you get the NNP minima, we take out some of these low energy conformers. Specifically, we take NNP minima within 25 kilojoule per mole range, and then we perform direct TFT. When we perform direct TFT, we also calculated for their harmonic frequencies. So this is essentially the vibrational spectra. So we also try to predict the vibrational spectra, but this is the scaled harmonic. So another way to calculate the spectra is via anharmonic calculations, but that's more expensive. So for now, we stick with the cheaper one. And if you haven't noticed already, the name of the game here is being cheap but accurate. Okay. So what you see here is actually the calculated global minima of each of these structures. And here I've only shown here the glucose, galactose, and mannose, and their corresponding methylated structures. Now I will be talking about the methylated structures later. So all in all, I have not only worked on the protonated and acetyl systems, but also their methylated forms. Now, why is their methylated forms so important, or why do we even have to care about their methylated forms? So, analytically, in an experimental setting, the unmethylated form of these sugars can easily adapt the alpha and the beta conformation, the anomeric forms, right? But what if we are, and I've mentioned earlier, the specific isomers, they can have different what do you call this interaction with, let's say, a peptide, right? So an alpha conformer will have a different interaction or binding with a peptide when compared to its beta form, for example. So it's important to identify or distinguish from the two. But if you have a solution of, let's say, glucose, right, you can have both the alpha and the beta. So how would you know, right? How would you differentiate? How would you be able to study each of them separately? And so that's where methylation can come in, right? Methylation allows or inhibits this sort of ring opening in order to get to the other anomeric form. And so in practice, most experiments, they do methylation in order to freeze this and then study them separately. Now, in the previous paper, the reference paper that we have used for this one, they claim that methylation doesn't really have a big perturbative effect, right? So that means if they look at the structure, or if they study the system of a methylated structure, they think or they believe that they're essentially getting the same behavior, structural behavior, as the unmethylated form. So they can just map it back to the unmethylated form. And from their experiments, they actually saw this phenomenon, right? And in this case, so we're methylating O1, right? What they found in their experiment is that this O1 just disappeared, but they still retained all the peak patterns and in this region here. And so they believe that even if you methylate it, it still retains the overall conformation, right? So that's an important that's actually a very important insight, because in the future, even if they methylate, well, we still preserve the same structure, so that's totally fine. Now the question is it actually applicable for the rest of the other isomers? Now, experimentally, that's quite challenging to do, because these are the other ones, aside from these three systems, are not readily available in real life. They had to synthesize it, and if you know synthetic chemists, it can be quite challenging in some cases. Particularly this one, it has N-acetylation, so there would be several steps in order to get this particular structure synthetically. So that can be challenging. But it is challenging, but in this case, we only want to know whether it actually has merit to look into whether methylation can have this significant effect. So that's where we come in. Since computational, or at least the computational approach would not require this synthesis of these systems, we can just easily simulate them. But I say easily, but we know it's going to be very expensive if we don't use NNP. But since we have NNP, then we can do it. And so here I'm showing you the I'm showing you actually this one that I had mentioned. So when they methylate it, this is the only peak that disappeared because O1 is being methylated. So the OH stretch is disappearing here. That's the expectation. This is the ideal case, right? But can we find the same behavior if we look into the other isomers? So actually what we found is that although most of them followed the same trend as this one here, we found 2 cases. 2 out of the 16 cases wherein the global minima actually changed its overall conformation. So for example here, this beta altrose N-acetylated, initially it's a chair conformation from its unmethylated form, but after methylation, it changed to a skewed boat conformation, right? And the other thing here is that this beta thallose here also changed its hydrogen bond network. So what do we get from here? It means that in most cases, yes, after methylation, you will still retain the overall conformation, but caution must be taken because 2 out of the 16 cases, or 2 out of the 16 isomers have shown that this can happen, like the change in the overall conformation can happen. So again, we were only able to find this out because we actually performed these calculations, right? And of course, we are not too unmotivated to do it because we have NNP. Okay. So what actually happened structurally? So for this structure, this is the unmethylated form. We can see here that this is the O1 stretch, or this is the O1 group, and it's actually the hydrogen bond donor to this O6 oxygen. Now, what happens when this gets methylated, right? If this gets methylated, this hydrogen here disappears, right? And so we're losing this hydrogen bond, and we know the overall stability of structure also depends, is also influenced by hydrogen bonding. Usually more hydrogen bonds, more stable. So now we're removing one hydrogen bond here, and so what happens? Now we change the overall conformation. From a chair conformation, it's now a skewed boat conformation, right? And our prediction here, we have this overall, there's a lot of things that happened here. Of course, O1 disappeared, but this O3 here become redshifted, right? So that means methylation here doesn't really work well, right? Another case here, the overall conformation, the overall ring puckering is still chair, but we have this change in the orientation of the N-acetyl group. So essentially, we get a totally different conformation for the global minimum, right? So again, these are the 2 cases wherein methylation can significantly affect the overall conformation. Okay, so maybe it's still safe to say methylation is okay. It will not change the overall conformation, but you have like 1 out of 8 chance of not being able to actually observe that, right? So it's not 100%, and that actually helps us as well as computational scientists because that gives us a job to do, right? We get in a sense we say that there is no particular rule that can be followed 100%, right? But only through our calculations, only through our explicit calculations that we can point out these inconsistencies, these discrepancies. Okay, so yeah, I've already mentioned this one. So Have questions so far for this work? Or do you need
Okay,
this?
so as Ajanon is in the meeting, so yeah, I'm taking over as a MC for now, and also asking you some questions.
Yes, yes.
Yep. So, so it is about the overall speed of this
Yes.
process. So now I've seen that your robustness of training the model for training the model for the guidance, and if functionalizations no problem,
Right.
you can still
Right.
get the structures right, and also can reproduce the IRPs, right? But then like about how faster you're like running optimization
Mhm.
with NNP to just compare with the optimization using common DFT.
Right. Sorry, what's the question?
Yeah, how fast?
Oh, how fast? So the one that I've shown earlier, I said it's 100%. I'm sorry, not 100%, 100 times faster, but that includes all the processes, not just the calculation itself. But if we're just looking on the NNP versus DFT, for example, I think it's 1,000 times. Around 1,000 times. But what makes it less than that is because we still have to do some training. If you do DFT, you can just do it directly, right? But for us, we still need to do some single point calculations and the actual training, and that would also depend on which hardware you use for training. So you can do the training on CPU, which is extremely slow, or use a GPU, which is considerably faster, right? So yeah. I have a question.
As the sampling has been done with like 3,000K
Yeah.
MD simulations, right? So does that mean that if we want to do something like ab initio MD but using NMP potential, is it going to be so the robustness of this model still work, and also to simulate the reaction,
Right.
the reaction pathways, maybe the whole or maybe like the whole process of methylation and
Yeah.
and finding the methylation and finding like let's say the pathway
Right.
of methylation.
Right.
So can you also do that in principle?
So short answer, yes, but there's a very big but there, because that means you now have to work harder in training, right? You have, if you want to cover this pathway of, let's say, methylation or reaction pathway, then you would have to sample transition states, right? You have to sample. So the sampling complexity would always depend on the type of system, type of process that you want to explore. For us, we only looked at the low energy region, so that means we can cut a lot of corners. Like we can remove the high energy conformers in our data set because we don't really need them in our training. But let's say, for example, I want to use this NNP in order to run molecular dynamics, even at the gas phase, for example. Then our training set would have to include some of the higher energy conformers as well, even the slightly elongated bonds, for example. So it's not a clear cut that, oh, you have an NNP that is trained for this particular system, then you can immediately use it. You still have to assess whether this model can really do it. So usually we do, like, for example, we do molecular dynamics sampling, and then we test the data on our NNP. But most of the time, if the NNP is just trained on the low energy structures, it will break. It will not be able to describe it. Yeah. Good.
Thank you.
Should I continue?
Yeah.
Oh yeah, please.
And-
Oh.
So you mentioned about methylation that can actually alter the conformation of the glycan, and and if you don't methylate, then the like conformer will flip all the times, right?
It can.
So how actually can you assess whether you are like compute IR is actually accurate if methylation is not working.
Okay.
So do you still need methylation, or you need to do some like maybe freeze the molecule or something?
Oh.
Or you have alternative strategy to prove that your calculate is good?
Okay.
Thank you.
Yes. So actually, to clarify, for us in computational side, we don't really need to do methylation, right? We are doing methylation because we want to help the experimentalists to be able to recognize that, oh, this structure is actually the beta form, this is actually the alpha form, for example. But in this particular work, what we wanted to show, or we wanted to confirm whether the claim from the experiment, because in the experiment they have done the unmethylated and methylated form, and they have claimed that methylation doesn't really alter the overall conformation of that particular sugar, even if it compared to its unmethylated form. But here, what we have shown, and that's the reason why we have also done methylation. But the advantage for us in the computational side is that we can prevent the flipping of the alpha and the beta conformation, because this is calculation. We have a lot of control over it. And so in some cases, actually not in this work, but some experimental groups, they don't have to methylate. What they usually do is they run it through ion mobility spectrometry in order to separate them, right, without even methylating them. And actually, not in this work, and not the one that I'll be showing you later, but we were also able to show that. Oh, sorry, we were able to identify that the one that they have separated is the alpha or the beta conformer from our calculations. Because in the experiment, although you have separated, for example, these two alpha beta, you don't know which one is the alpha, which one is the beta. So that's where we can come in, right? But to answer, going back to the question, we don't need to methylate, but we methylated it because we want to address this particular question that we have based on what we have seen in the previous paper. Yes. Okay, so I think I need to finish the rest of the presentation. Okay, so the next part. Now, in the previous one, we worked on protonated N-acetyl hexosamines. Now, are we really just limited to protonated structures? And actually, in fact, protonated structures or proton in general forms a covalent bond, and so it can be more perturbative to the actual structure of the neutral form, right? And so we have just actually submitted our response letter for this work that we had submitted to BCCP, and here we actually explored, rather than protons, we looked into alkali metals. So we have lithium, sodium, and potassium ions. And then we look at, this is the same N-acetyl hexosamines, and we look at how the conformation changes, or is it still possible to differentiate these systems using these ions in terms of their vibrational signatures. Okay, so this is still the same similar process, but here the pattern transfer that we have used, aside from the flip and rotate, is just the direct substitution of the ions. So for example, we have a database for sodiated structures. If we just want to cover, or we want to sample the lithiated structures, we just directly replace sodium with lithium. Now, the new structure, the sample structure, of course, will not be in its most stable state, because, of course, there's a change in the charge density. And so we still have to optimize it. But again, the idea is that it should not take too many steps to re-optimize to the lower energy. All right, so now we went a bit more, what do you call this? More ambitious. Because before I've shown you I've only worked on 32 systems. Now we kind of doubled that, because we have NNP. We kind of like trying to push the limit of NNP. Since this is quite fast, why not just work on more, right? So now we have covered not only the 3 ions, we also work on the neutral and all their 8 isomers.
Okay, so in total, we have 64 systems at the DFT level. Okay, now we have collaborated with the Japanese group with this one headed by Professor Shinichi, and they have provided us a really nice high resolution spectra through IRPD, and this is under cryogenic conditions. So, well, they're very, I mean, they have a lot of resources to do this. So, we were lucky to have collaborated with them. And so, this will be our basis. We will compare our calculations from these high resolution spectra. Okay, so we have worked on only 3 at least. We have calculated all 64, but the experimental data that's available is only for the 3 systems, glucose, galactose, and mannose. So I'll be showing that here. Now, when we look at the galactose system, okay, it appears that, so the green here is the experimental data, and the blue one here, right, is the global minima that we have calculated. And it appears that apparently, okay, only the using just the global minima accounts for all the observed peaks. Okay, so what does this mean? This means that, yeah, the observed spectra here is only one conformer. Now, it just so happened that, so this is lithium, this is sodium, and potassium. All 3 of them showed very similar structures, right? You see this one here, this is the only difference, right? It's like being blue shifted as we increase the, as we decrease the ionic charge density of the ions. Okay, and if we look at their structures, this is actually how it looks like. So this is a smaller one, lithium, sodium, and potassium, and their general conformation stays intact. Okay, so this appears to be very simple, right? If this is also applicable to the other sugar systems like mannose and glucose, then that's really fantastic, right? That means that we don't even need to do any more calculations. We find this for galactose, we say, for glucose, it's gonna be just the same, right? But since I'm already saying that, that means there's a twist, right? So when we go to glucose, now it becomes a little bit more complicated. First, the observed spectra doesn't only account for the global minimum, right? In fact, we had to use, just going back, these are all alpha conformers. So in here, in the experiment, they considered both the alpha and the beta. They did not separate alpha and beta. This is just one solution. It can be a combination of alpha and beta. But we found here that only the alpha global minima can account, or is enough to account for the observed spectra. For glucose, we find that still only the alpha conformers, but we need 2 of the lowest energy conformers to account for the observed spectra. And actually, they are not entirely the same. Because here, I have color-coded this based on the coordination and the ring puckering. And so same color means similar structure. So this one here is similar to this and to this. And at the top would be lithium, the middle is sodium, bottom is potassium. And just to give you a short summary of what happened here, we needed 2 conformers to account for the observed spectra, and they're not entirely the same. I mean, they're not similar at all with each other, because the first one here, the global minima is the blue, but for the sodium and potassium, the global minima became magenta. I think it's magenta. Yeah. So there's a bit of a shake-up in the energetic ranking in the trend. So from here, we know it's not as simple as the galactose, right? Now, how about for mannose? How crazy could it get to mannose? Now, mannose, this is where a lot of things have happened. A lot of crazy things. First, for lithium, okay, first 2 lowest energy conformers are the most, account for the observed spectra. But for sodium and potassium, right, we now have to use more conformers to account for the observed spectra. And the other thing is that, I know you guys cannot see it clearly, but for the lithiated system, both the alpha and the beta conformers are needed to account for the observed spectra. So again, we go back, galactose only global minima of the alpha conformer. For glucose, we need the global minima and the second lowest energy conformer of the alpha form. But for the mannose system, both the alpha and the beta conformers are needed in order to account for the observed spectra. So it really gets a lot crazier. And actually, we have found, actually the reviewers have seen this and asked us to look more into it. And we have seen some evidences of kinetic trapping. Now, if you're not familiar with kinetic trapping, this is a phenomenon wherein a relatively higher energy conformer can get trapped in a specific, can be ion trap in the process, and that they can be observed, they can be measured by IRPD, right? Although, for example, here, this is 10.8 kilojoules per mole. This is relatively high compared to the other ones, which sit at around, this is, I think, 4.1, 5.4, 3.2. Yeah, this is relatively higher energy. And actually, in between here, there are other conformers as well that we have calculated. But it so happened that in the experiment, apparently, this structure here could have been measured, right? And that is what we call kinetic trapping. And so this kinetic trapping happens if the well where it sits has a really high barrier, energy barrier, right? Usually it happens when you have coordination with a lot of atoms, because in this case, this is 4 coordinated ion, right? And the same thing happened with potassium, with mannose. Okay, so what's the message here? Well, the message here is that there's no simple rule to really cover everything here, right? There's no simple structural behavior that can really capture what we could expect from these methylated systems. Galactose seems to be the easiest to account for, but unfortunately or fortunately for us, we have some jobs to do, right? Now we cannot just say, oh, we calculated this, then the rest you can just predict that it will go this way. But no, right? We still have to look into those calculations. We still have to explicitly say or find out what the structures would be and their behavior. Okay, right? But at the end of the day, we still see that these ion systems can still be used to differentiate these sugars, at least the 3 sugars that we have looked into the experiment. But I did not show it here. We don't have a lot of time, but even those that are not glucose, galactose, and mannose can be differentiated. At least we predicted that can be differentiated via cryogenic IRPD. So there's a lot other, there's more things to this, but we don't have a lot of time to discuss it. But at least this, I think, what's the important message that we want to put across with this project here. So hopefully it gets published soon. Right. So I'll be open for your questions.
Okay, so any question from the floor before I do? No? Okay. So now I would like to discuss a little bit more about the ion kinetic trap. Yeah. So for the calculation and also I'm not like I'm not an expert on this, but for both the calculations and for the high resolution IR, so
right.
has it already included like all the possible sites for ion trapping? Like it could be trapped in the conformation shown, but I believe that the ions can also be trapped on the other sides with different probability, right?
Right.
So has this already like been accounted in the calculations?
Okay.
So actually, this is actually one of kind of like the questions by the reviewers when we have submitted this, and in particular, they're asking about the barriers of these conformers, right? But to answer you straight, we do not calculate any barriers. So this is more of a speculation for us based on what we saw in the structure, because here, where we kind of observe kinetic trapping, all of these other low energy conformers don't have four coordination systems. Only these that we suspect that are kinetically trapped have shown this four coordination systems. So even though their energy is relatively high, if the barriers around them is also high, then it gets harder for them to go out. And the other thing is that we asked our collaborators, and they said that they did not really do a thermalization step. So thermalization would try to kick out these systems kinetically trapped to get to the thermodynamic equilibrium. But if we don't do that, then they can get easily trapped. Especially this is low. This is a low temperature experimental setup. Yeah, but of course I'm not an experimental expert, so I cannot really say to a more detailed aspect. But we believe that's what happened. At least our collaborators have also agreed with us. So yeah.
And also about these IR, like to that you can benchmark your IR prediction with the high resolution IR from the experiment. So what's the IR predicted by your model came from the training based on the BFT result, but is it still somehow reproducing high resolution IR in the experiment? And how about if we if we just compare the direct DFT that like IR peaks from the direct DFT with high resolution IR, is it kind of different from this?
Right. So I think I have to clarify. So these calculated spectra, these are DFT, not NNP. So we did not do any training on the NNP. Remember, our NNP is only trained on the energy and atomic forces. So we did not train on dipole moments or anything like that in order to predict. But that's actually a good question, because this is also one of the things we're currently working on. Either we look into training the image of the spectra, or we actually train with the dipole moment of the structure, because we can do velocity autocorrelation calculations in order to get actually not just harmonic scale but the unharmonic effects as well. Hopefully we can do that in the future. Yeah. Oh, good. So I think I'm okay.
Okay.
Right. That's everything.
So let us thank Kenny for the very nice and intuitive talk. Hopefully we have many things to do in the future. Okay, then, thanks Kenny again.
Thank you.
Okay, and then I think that we would have like a short break. Okay, so there might be
Hi, sir.
there should be some breaks on this provide, and yeah, that gives you maybe some times to wash your face and go to the toilet like for 5 minutes, where we are preparing the slide for the next session by Professor Brad. Okay, okay, so it's like 5 minutes. Do I use my phone? Yep, that's good. So I don't need my computer. Yeah, I need you can use this one. Already installed this slide.
Graph demonstrate the optimization.
Just update one page.
Compared
Okay.
Can I borrow the picture?
lower energy. So which one is the
This one?
yeah? The blue one is you got from the real experiment.
Oh, no, it's the green one.
The green one.
Yeah, the top one. Yeah.
Is for you got the molecule energy rate.
So the green one is the experimental data that from Japan, and then the other colors are what we have calculated. So-
based on optimization.
Yeah, so the optimized structure, right? We can do you can do frequency calculations. You don't need NNP for that. So you just need to run NNP calculation with, let's say, if you have Gaussian, any software can do that, and then they can generate something like this.
So
Yeah,
you screen, you got 1, 2, 3, 4, 5, 6
yeah.
potential option and compare.
Oh, yeah. So what we actually have, like in our calculations, we can have a lot of conformers, right? but we only showed here the ones that we think are like they're matching the peaks, because what we want to do is like they should match, so that if they match, that means, oh, this particular structure is in the experiment. Yeah.
It's a conformer, but
So how are you?
the
Good.
way
So you just traveled this morning?
way
No,
high of the way
yes.
and
Mhm.
precision move is the work.
In this case, it's the what do you call this? The height in the experiment can mean different things, but one of the most direct ones is that it could refer to the relative population, right? Or the probability of finding that particular conformer. The position would depend on the vibrational mode. So, for example, if you have a loose bond, then it will have a lower frequency. If it's a tighter bond, it's harder to stretch, then it would be on the higher frequency. Yeah.
So you mean that your optimization can be possible to
Oh, yeah, yeah, yeah. Sorry.
to that one way similar performance?
it was kind of the same way. Just like different. Okay, so can you please put this on your
Oh
sleeve?
yeah,
So it's actually on the
for voice recording.
yeah,
I know,
voice
I know, I
recording.
know. I have a administration team is doing exactly the same thing. This is I got a new job. I'm in charge of digital courses of the whole university. So we do this
Wow,
every day.
every day.
I have a couple of
Seems like a
staff member
also
recording, broadcasting, and
lectures.
yeah. So try to help the professor to
And
do
it seems
this.
like the whole NTNU, right? So
That's the for whole university.
whatever course they will come
Yes,
to you.
yes. And we have a platform. It's a Moodle platform. It's an open source platform. So
So that's why maybe you might get like busier, busier.
yeah, I have been
with these
super
things.
busy. Yeah, I keep just sending message, writing emails. Yeah. But it's good because this kind of stuff relate to AI.
Mm.
Yeah. So I think that's kind of a extend from what I study, a little bit to the education perspective. Yeah. Especially for my talk, you can see I prefer to use text. I don't prefer physical information, meaning structure. I prefer text. Text is kind of a way human communicate
Mm.
that actually embed a lot of information. So I think this is correlate to what I have been studying. So I think it's fun. Yeah. So maybe you can put another direction to this way.
Yeah, actually, you are in the same position as similar position as Non. Actually, Non is
Yeah.
the
I know.
director, but for this faculty.
Yeah.
So now, actually, luckily that we have his staff here.
Yeah. Yeah. It's fun. So how long should I talk? 80 minutes or 90 minutes? Oh, excellent. How long should I talk? How long?
I'm sorry.
How long should I present my talk? 1 hour?
It's okay.
Maybe 1 hour.
1 hour?
Yeah, 1 hour is good.
Roughly.
Well, it's good. Okay. Including QA or not including QA?
Whatever is
Oh, ever. Okay.
Yeah, and
Flexible.
when we finish, we just go out.
Okay. Sure. Do I need to away from this camera or
อ๋ออาจารย์ครับ
is this working?
ตอนนี้ปิดอยู่แต่ว่ากล้องเขาถามกล้องนี้ทำงานไหมบอกว่าตอนนี้ปิดอยู่ครับแต่ว่าเวลามีคนถามอย่างเงี้ยจะถ่ายไว้ที่คนถาม
Oh, that's for filming the.
Oh, I know. I just want to make sure this is also filming. So if I should
บัง
stand.
บังได้ครับบังได้
Oh, it's okay. It's okay.
It's okay.
It's okay.
If I do this, it's okay.
Because it would open just only when questions.
Oh, okay. I see. That, yeah, that's why I'm asking. Yes.
So อันนี้คือผมก็ถือไปได้เลยใช่ไหมครับ
ใช่ครับ
อย่างนี้เลยตอน
อัน
นี้
นี้อันนี้คือเสียงมันเข้าแล้วนะ
เอาสิครับโอเค
yes so it does this
Is it live broadcasting on YouTube?
It is.
Live.
Yeah, but it's just like for like faculties.
I'm
in
okay.
universities.
I just want to make sure it's live. It's live. Okay. If it's live, that means there's no room to talk about anything. Yeah, we usually do not live because we want to keep the freedom to edit for the video. Yeah. Because sometimes people go too far, especially when they talk something more topic away from science. Yeah, that's what we do. So
อันนี้คือไลฟ์อยู่นะ Okay, so now we are going to start like the second session. Okay, and it's a pleasure today to have Professor Brad or Min Kang Sai from NTNU Taiwan. Okay, so he would be talking about also the application of CHNET. Okay, so we have been with CHNET for the whole workshop, right? And then it was pretty nice thing that CHNET can predict. You can train CHNET to predict and calculate like physical properties of small molecules like peptides. Okay, and also it can predict some of the experimental like IR, right? But here for this talk, I think it would be about like the more aspect that CHNET can do. Okay, so it would be more of the general purpose, and I believe that this talk would inspire many of you, okay, that find something to collaborate AI, okay, applications of AI with chemistry and all. Okay, so now please welcome Professor Brad, okay, for his talk.
okay, thank you. Thank you, Professor Tana, for the introduction. It's great to come back here again. My name is Brad, coming from NTNU. I'm a professor in chemistry, and I'm also a joint professor in Tsinghua for the Biomedical Artificial Intelligence program. And in addition to that, actually what I'm doing these days highly related to this topic. The topic today is about how AI can be applied to chemistry. So there's a two key domain. One is chemistry, one is AI. So right now I'm also helping NTNU to building petaflop supercomputers. In order to run AI, you need to have good supercomputers. So that's one thing. That's the instrument part. And my another office is doing online courses for the whole university. So we manage the teaching platform. Whoever the faculty want to do digital teaching, they can collaborate with us. So I think it's the same thing as what we are doing the YouTube broadcasting right now. Yeah. So the last one is my research center. That will be the main result given by for support today's talk. Okay. I'm going to talk about how we look at the molecule or the material from the very beginning. This is called representation. By given a more quantitative description, people can represent the molecule from different ways. So if you write a molecule in terms of this chemical formula, it only can teach you the molecular weight and the number of atoms if you write this way. So it's a very limited information embedded in this written part. Okay, so let's define this as 0 dimension. And you can also extend the molecule if you write more complete chemical formula. In that case, you can identify the functional group of the molecule. Okay. So in this case, for the example of this molecule, it has amino group. It also have a halide group. It also have aromatic ring. So you can use the presence of this functional group to represent this molecule. So each bit here represents one functional group, and you can expand the number of bits to do a complete description of this molecule. So we call this one-dimensional representation. Okay. So these days in machine learning, we call this fingerprint. Okay. And you can go beyond this to do the connectivity of the molecule, where you can use a matrix. Each row is the sequence of atom, and the other direction has exactly the same sequence. And you can describe, does this atom connect to the other atom through the number of bonds? Okay, so you can do this way. So you have I'm going to show you the real matrix in the later slides. So it's a 2-dimensional array. You don't actually need to know the 3-dimensional structure, because we write Lewis structure and we know how atoms is connected to each other. So we can count the number of bonds connect between any 2 atoms, and then we can store this information in the matrix. So it's 2-dimensional. Dimensional matrix, that would be the fundamental information provided to ShredNet to do the advanced machine learning application as introduced by Kenny earlier. So because it's a physical infused information, the distance has very precise decimal numbers between atoms. But in order to have that information, you either need to do first principle calculation, or experimentally you need to do crystallization. Otherwise, you don't have this. Okay, so we call this 3-dimensional representation. And you can go beyond this, called 4th-dimensional. The 4th dimension, that could be the change in respect to time, or the change of, for example, electronic structural information projected on each atoms. So it depends. The 4th and 5th and 6th, you can go unlimited dimension. But the principle is, if you add more information, you need to prepare more. Okay, so there's always the balance. How much effort you want to prepare for your data, and do you really need that in order to do this application? So there's no absolute answer for this. This is just the balance between your motivation and your effort. Okay, so I want to define this called molecular representation in the very beginning. So the topic I'm going to talk about is a photophysics property of the molecule. That's a very typical and a common property for the molecule. So in order to describe a photophysics property of a molecule, usually people use this Jablonski diagram, and that's a way of drawing the ground state and the excited state of the molecule. So if we know the ground state of the molecule, and then we can define the energy level of the ground state, and then we can do the excited state description. In that case, the energy difference between ground and the excited will be the light absorption from the photons. And from the chemistry, we know when the molecule is excited to the excited state, it can go some interconversion between the singular state to the triplet state, and we call this intersystem crossing problem. If the molecule is relaxed from the excited state down to the ground state, and usually experimentally we can observe the fluorescence wavelengths. So that's something the experimentalists can probe. And when you pump the molecule, we also can measure the absorption wavelengths. So that's also the experimental information can collect. And if it undergo the intersystem crossing, and then it can react to the triplet surface, and some molecule is capable to release another photon. It's called phosphorescence. But the fluorescence and phosphorescence, they are both involved in light emitting process, but the time scale is a little bit different. So experimentally, they rely on how the instrument can distinguish these two processes. That would be complicated anyway. So from the computational point, building one diagram can only be used for one molecule. So if you want to describe one molecule, its excited state information, you just build this diagram. And these days, density functional theory is well capable to doing that. But the problem is just one diagram for one molecule. How if we want to do 10,000 molecules? That you can imagine how much effort you need to put in this. Okay. So the way we are working on the problem like this is we know from the experiment in biological application, the fluorescence protein is well known in these days. There are many animals or fishes, they have colors that also involve the fluorescence protein in their body. And the biologists, they know how to change the fluorescence protein sequence, and they can tune the color. So these days you can see, you can design the color if you want. You just change the amino acid sequence in the fluorescence protein. Okay, so there's a relationship between the color and the sequence, or more precisely say, there's a relationship between the color and the chemical molecular structure of the fluorescence protein. Okay, so we are inspired by this. Can we just build a quick machine learning model to connect the structure and the color? So that's the goal we want. Okay, so in order to do that, we go to a commercial data set. This is called a ReACI database. It was, I think it's provided by Elsevier. We download this data set from its database. So it contains around 11,000 organic molecules. Those are already be synthesized by the experimentalists. So they are the molecule exist in this world, and they has been measured by the experiment. Okay, so you can see their distribution of the experimental, the maximum emission spectrum of each molecule. That's the very typical distribution of this data set. Okay, so the next one is, can we represent this? So this is the project this is our first project. I think this is seven years ago when my first project moving to machine learning. Before that, I'm like a 100% first principle guy. Okay, so we decide to represent this molecule. So we quickly check the idea of represent a molecule in the chem informatics community is usually called descriptors. Descriptor is just a sign to describe part of this molecule. And there's many ways of building descriptors. Okay, so this is the descriptor generator we try, and this is open source. And so meaning if we fit this molecule, use this SMILE file to represent the molecule, and then we fit to the descriptor, it will come out with about 11,000 description about this particular molecule. It's like you met a new person, you want to memorize this person, and what exactly you memorize? Maybe his gender, maybe his hairstyle, maybe does he wear glass, something like this. Those are fragment information you memorize in your brain when you meet a new person. So you do exactly the same thing for the molecule. So descriptor can generate this fragment information. But some information is important to the property you are interested in. Some information is not important. So the tedious way is how to get rid of those not important information. Okay. So in order to do this, you need to do descriptor dimension reduction. So get away from those unnecessary information. Okay. So let me go to next slide first. Okay. So the way we do this, we as I mentioned earlier, we have 11,000 molecules. Each molecule have 11,000 descriptors. Some are important, some are not important. So the first stage is we do the unsupervised clustering of those descriptors. We want to categorize those descriptors to see, can we somehow find some hint to get rid of this, okay? So the way we are doing this, we implement this regularization penalty function, and then we clusterize by the 11,000 type of descriptor into 15 category. The determination of the 15 is based on the scoring function. So you can check, there are many scoring functions you can pick. So among those 15 categories, we pick the top 2 or 3 descriptors and then assemble back to form these 480 descriptors. So that's what we do through this unsupervised clustering technique, okay? So you can recognize now the descriptor space is reduced from 11,000 plus to 480, okay? You can even make it smaller if you want, okay? But if you use fewer descriptors, your prediction error could be larger. So it's also a balance. Okay, so here we have 480 descriptors. Now we go back to use the regression method with those 480 descriptors to do the prediction. And this would be a very common regression method these days. It's very powerful, I have to admit. This is super powerful. You don't actually need neural network to deal with every problem. Okay, so it's called random forest. The random forest is it randomly pick up the descriptor to split the data. Okay, and so the data, depending on the criteria of the splitting, it pick up by the problem. For example, a typical example is if I just split the data by the molecular weight at 500, you can pick this. Molecule bigger than 500 with its molecular weight would go this way. The other rest go to this way. And then you go to next layer, pick up another. You continue to split until you reach one molecule at the final, one molecule per node at the final layers. Okay, just keep splitting. And then you construct one tree. And then you ask the program, can you do more tree for me? Because there's multiple combination of splitting this molecule. Okay, for typical case, I'm going to show later, we usually construct 100 trees. And then use those 100 trees to predict the property. So basically, the program, it just memorize all the splitting of this data set. So that's what program was doing. Okay, so we have a 480 descriptor and we do random forest regression. Now we can predict the property we want, okay? So this would be the prediction of the emission wavelengths of each molecule. And this is the error bar of our prediction, okay? And of course, you can use linear regression. Linear regression is commonly, we taught this to analytical chemistry laboratory. All students know linear regression. Linear regression is not bad, but the linear regression will give you the contribution of each terms because linear regression has very precise form, okay? Random forest has no precise form. You can not easily to interpret what exactly is random forest. That's the problem. So we use the same data. We test the linear regression. That's its error bar. 44 nanometer for predicting the maximum emission wavelength of a particular molecule. So at this stage, it depends. Do you think this error is small enough for you to design your next step for the experiment? If it's good enough, you can just do with linear regression. It's okay. Okay. But if you think you want to do better, you can switch from linear regression to random forest. And the error can go down from 44 nanometer to 36 nanometer. Okay, so the lesson is very clear. You go more complicated model, your error also go smaller. Okay, but your effort go higher. So it depends where you want to stop. Okay, because you decide the final application. So people always has to think, what I'm going to do with this model. It's not just doing a good model, it's what is model going to serve me for what, okay? So that's the key message, okay? For random forest, you can actually extract this called feature importance. The idea of the feature importance is that because when you split the model, you pick up the criteria of the splitting. So some descriptor is picked within some trees. So you can count the number of times this descriptor was picked for building the tree. That shows the importance of this descriptor. And then you construct 100 trees, and so you can do a statistic in a certain way to describe how important is this descriptor. So in that case, we identify from those 480 descriptors, the first one, the most important descriptor is the number of conjugate double bond in a molecule is critical for predicting the wavelengths. This is consistent with chemistry, okay? We know when you have the more number of double bond in a molecule, you are going to make the homo-lumo gap smaller, okay? That's what we told in the organic chemistry. So actually the random forest found this, okay? So this is always good. If you can extract something from the model and then compare with your domain knowledge, that will be critical. Otherwise, you don't know what exactly its model is doing, okay? So this is the first lesson, and then that's our first try. Okay, so now the problem is that, as I mentioned earlier, when we construct the data, we just take the SMILE file of the molecule, fit to the descriptor generator. When we do this, we ignore the solvent effect. Because in experiment, if you predict the experimental property directly, there is always environmental effect. That could be the solvent, that could be the temperature, that could be the pressure, whatever, okay? But if you just take the molecular structure, use that as the initial information and to train the model, that means you assume the solute contribute 100% to the property you are interested in, which is not true when you predict experimental property. So if you see a paper, they predict experimental property, just use solute information. Okay, you need to be aware that they ignore something, and is it really making sense? Okay, so we want to examine our approach in the first task. So we switch data set. The previous task is commercial data set, experimental property, and then we want to confirm is our methodological development really is rigorous or not. Okay, so we switch data set. We pick this theoretical data set. This is CoQM9. It contains 134K organic molecule computed by first principle calculation, which is DFT. So in that case, it was computed at this level with this basis set. It almost consists all the possible molecular structure within 9 heavy atoms. Okay, so this is a public available data set. You can go here to download this. Okay, so in this case, this data set has no environmental effect. Okay, and this is no human effect because it's just complete by one research group through computing program and then keep generate the data. Okay, so we do exactly the same procedure as I mentioned earlier. Generate descriptor and then do descriptor dimension reduction, and then do linear regression and the nonlinear regression random forest. And then we check the error, okay? So you can see here, we predict 3 properties. One is the energy level of HOMO, energy level of LUMO, and the HOMO-LUMO gap. Those are highly related to experiment, your oxidation potential, your reduction potential, and your absorption and emissions. So they are correlated, but they are not exactly the same thing, okay? So that's why we want to test the method. So you can see here, when we do the linear regression, our error is 0.161 EV, okay? Convert to kcal, that would be the 4 kcal. So we have 4 kcal error to predict the HOMO related to oxidation potential. Okay, and then for the LUMO is around 5 kcal for the reduction potential. Okay, so that's how we get confidence about our methodology, because we need to know how big is the error. Otherwise, we cannot imagine what application we can do. The HOMO-LUMO game will be a little bit tough if you do the linear regression. Okay, again, you can switch from linear regression to nonlinear regression. That's random forest. Here you can see the error is reduced. Of course, you go from linear to nonlinear. The formula become more complicated. Of course, you can get smaller error, okay, but not big. Just a little bit change, okay. So it depends. Does this good enough for you? If it's good enough for you, you can stop here to do application, to do the design of the new molecule, okay. Okay, so the next one is now we are confident about the methodology thinking. So we collect information, we reduce the information, and then we do regression. Okay, 3 stage. And we know how to collect it, and we know how to reduce it, and we know how it can match with different regression model to do the prediction. So that's the learning lesson. And the next one is, can we go beyond handcraft? Handcraft meaning the first stage, we need to collect the descriptor. That's from some online descriptor package. So somebody already defined what descriptor can be generated in that package. So it's human handcrafted information. And we are trying, can we just ask machine to do this? Ask the machine automatically to generate those information. So that's why I'm saying here, the going beyond handcraft descriptor, try the data-driven descriptor. So the descriptor is generated by the data and by the machine. So in that case, that would go into the category of the neural network. This will be the structure as Kenny mentioned in the many wonderful lecture earlier, okay? So I'm going to just quickly go through this and skip this, okay? So the key is this model need the interatom distance, and it also need the atom type of the data. Okay, so you need to know the condition, otherwise you cannot use this model. Okay, so that's the drawback. But that also give the power of this model. It can predict very precisely, especially for neural network potential. Because potential, if you have some error, that means the dynamic you are observing is totally garbage. Okay, so that's just a balance. Okay, so we are thinking, can we replace this interatom distance to do a quicker way? But of course, if we replace this, that will give you more error. Okay, that's the point of my next slides. Okay, so we're proposing we do bound stat instead of interatom distance. Okay, this is a typical example. This is a benzene molecule. If you have the Cartesian coordinate of this benzene molecule, you can calculate the interatom distance between each pair of the molecule. Very precise, 3 decimal, 4 decimal, whatever. Okay, but if you cannot afford the Cartesian coordinate, can you still do that? Okay, that's our idea. So here I transform this blue matrix to this red matrix. And the same molecule, the same sequence, but here I only counting the number of bonds through the valence bond. In that case, I don't need to know the Cartesian coordinate. Okay, I just write down the SMILE file or the Lewis structure of this file and ask my student counting the bonds. And I can construct this red matrix. So for this paper, we did not actually change the RAND model. We just fit the model different data. That's it. Very simple, okay? But by doing that, we no longer require the Cartesian coordinate anymore. That would be critical if you want to design something new. If you have a certain category, never be synthesized. But I have many organic colleagues or inorganic colleagues, the experimental colleagues. They have so many ideas. Give them a pencil and paper, they can draw new molecules, right? But if those are new molecules, that means no structure. Unless you calculate it. If you don't want to calculate it, how can you predict the interatom distance? That's the problem, okay? So the motivation for us to design the model is to provide a quick tools to my colleague. If you want to try something new, just write down the Lewis structure to fit, and then you can give you a quick prediction, okay? So by doing that, this will be the prediction for the bounce step matrix, okay? Here, this column is for the traditional way of doing interatom distance using Cartesian coordinate to provide the interatom radius as the input to the Schreiner model. Of course, it gives very accurate result. The HOMO energy is almost 1 kcal, and the LUMO and the gap is around 2 kcal. It's the state of the art these days. You cannot get more accurate than this, okay? But it needs the Cartesian. If you switch from the Cartesian to bend to the bounce step, you just double the error, 2 kcal, okay? So it depends. Is it good enough for you to use it? Okay, if it's good, then you can do this, still with ShareNet, okay? Just fit different things, okay? Okay. So we provide a high throughput tool to my colleague, and now they have become more greedy. They want more complicated property predictions. Can you predict quantum yield? Many synthetic group, they care about quantum yield because that's a real application part. If you can have very optical property for the future application. And quantum yield is very challenging because quantum yield is a very complicated phenomenon. The molecule go from ground state to excited state, and then through relaxation go back to the ground state. It has multiple channels, and the quantum yield involve molecular vibration because that's how its excited state energy is dissipated into different degree of freedom. So that, at least from the domain knowledge, that tells you probably need some information about vibration in the model. Otherwise, you probably have no chance to pick up this to predict a good quantum yield. Okay, so that's how I learned from this study. So molecular vibration from the domain knowledge. Before you begin constructing the model, you already know you need vibration because that's chemistry textbook teaching. Okay. So we have to change the Schreiner model because we know we need to fit the vibration into the model, and also we know if we want to predict the experimental property, we need to include solvent because that's how the experimental data was measured. Solvent effect and vibration to include in this model. So we modified the Schreiner model. This is the original Schreiner. Again, here it was interatom distance, that's R, between any pair of atoms. We switch that from bound step, it's okay. So the other one is we introduce solvation effect in this neural network working flow. But our argument is, if you want to introduce the solvent effect, there are multiple ways you can introduce this. You can add the solvent effect at the very beginning, like what you are adding the bound step here. So you can draw another arrow here. When you draw the arrow like this, that means the neural net, when it learn from the data, it would consider solute and solvent at the same time. So when you draw an arrow like this, this is the interaction layer. That's how those data was interacting with each other in respect to the final predicting property error. Okay, so that's how it adjusts. But if you put the solvent here with the bound state represent your solute. So if you put them at the same stage, that means those contributions are simultaneously considered and adjusted by the error. Okay, so that's the learning process. And we think this is wrong. The reason is, for photophysical property, the main contribution is solute. Solvent is a minor contribution. So we don't want the solution effect to interact with the solute at the very beginning at this layer. So we cut this and move the solution effect to the end after the solute contribution is determined. Okay, if you want to make an analogy to this, it's like you are solving the DFT, the self-consistent field, and you can turn on the solvent effect and turn off the solvent effect. Okay, you can always do a vacuum calculation and then add another solution, solvation correction. That's what you can do these days. So I just borrow that idea. I want to do vacuum first and then solvation correction. Okay, so in order to represent this error go all the way here. Okay, so that's how I introduce the solvation effect in the model. And the second one is, as I mentioned earlier, the vibration. The vibration is typically known, and in the physical chemistry textbook, if you write down the vibration, the vibration, the K constant, it involves the reduced mass. Because the vibration, if you look at more locally between a pair of atoms and bonded with a spring, and the spring constant is determined by the reduced mass of those two atoms. Okay, so we follow the idea. We use the molecular sequence, the molecular number, atomic number to represent the molecular atomic weight. So you can see the formula here looks a little bit similar to reduced mass. The reduced mass is Ma times Mb divided by Ma plus Mb. That's M. And we can use atom number to introduce very similar effect. And then we call this reduced atom number representation. And then we take this as a multiplier to each bond step. So we kind of stretch the value of the bond step to include a little bit inter-atom correlation, okay? So that's how we do this. And then after that, we just do the same ShareNet training, okay? So in ShareNet, you can actually use the same logic. If you have an environmental effect, do this at the end, okay? It could be a molecule embedded in a carbon electron material. That's physical absorption, right? You can do this. But the point is how you represent that environmental effect. That's the key, okay? And you can fine-tune the molecular bonding interaction here. This is the bond step. And you can add more term here. This is just bond. You can add angle, dihedral angle, and so on. Even chiral angle, if you want, okay? You can just add here. Keep adding. And after the neural net is constrained, and it will trend through the error backpropagation anyway. Okay, so you can always play around this. And then the whole idea is just a feature engineering. So there's nothing new. It depends on the problem, depends on the application. You can change the representation of the molecule, change the representation of the environment, and you got a new model, okay? So that's what we did. So now we build a model, and then so we have to examine how accurate is the model. And then this is the experimental data set we are comparing. This was published by a Korean group, 2020. It has around more than 15,000 organic molecule, maximum absorption wavelengths, maximum emission wavelength, and the quantum yield. Okay, that's the error of their model. When they publish this data set, they also construct a model to do the prediction. Okay, so here, if you remember, let me go back. So we have a solvation effect introduced here. So in the design of this, we can turn on the solvation and turn off the solvation. If we don't want the solvation, we just cut this off, and there is no solvation effect. So in our model, we can turn on and turn off. So you can compare these two gray number here. So when I put so here, that means the model has solvation effect, and this has no solvation effect. And this is the prediction error. for the experimental property. If you look at the first column, this is the maximum wavelength of absorption. Okay, turn on and off the solvation does not affect the model prediction accuracy for the absorption. Okay, which should be the truth because absorption is a vertical efficient process. It has very limited solvation relaxation phenomena. Okay, so let support the design, the model. If you introduce the solvation at the very beginning, you probably will see substantial error here because the learning process of neural net, they artificially put too much weighting to the solvation effect. Okay, and then if you look at the second column, this is the emission. This is without the solvation effect. This is with the solvation effect. Substantially reduction, the error. Okay, and that also support the way we design the neural net is more reasonable. Okay, so that's how we convince the editor this should be the way he designed because the solvation effect is minor effects, not the dominant effect. Okay, at the end we just use the reduced atom number approach, and then we can get a better content yield. The yellow number here. Okay, so the lesson of this work is just you need to have a very inside domain knowledge of the problem you are dealing with, and especially the chemistry formula already discovered because in the neural net you represent everything by a formula. If you already know some textbook have very important formula or equation about this, you better look at that, and then to connect from that formula to the neural net. Can you just follow the idea? Okay, that's the lesson we learned. Okay. Okay, and then the next one is this is a minor project. We published this earlier this year, and then we recognize predicting with experimental property is very painful. Okay, this it has 4 experimental data set. These are 4 experiments we collect from the literature. Okay, these are the reference. And you can see here, when people do the experiment, they usually just synthesize one molecule, find a suitable solvent, and then make it dissolve, and then do measurement. Okay, that's a very typical operation procedure. But some molecule cannot dissolve in some solvent. That means that experimental data cannot be collected. So the solvent to solute ratio is always very small in the experimental data. I can show you here. For a typical data set like this data set, it has unique solute 6,000, and it has used 359 solvent type in this data set. So theoretically, combination should be this number. Theoretical combination should be this number times this number. That should be an ideal data set you want to use as a computational guy. But the reality is you only get this. And this only take 0.7% of all the possible combination. That's pain in the ass as a computational guy. Solvent is underrepresented in the data. Okay, right? So it's a real problem. Okay, so we check 4 data set, all the same. Solvent is underrepresented. So if your colleague asks you, can you predict this molecule in different solvent? You got into trouble. There's no training data. How can you guarantee your prediction is good? Okay, so that's the problem of this paper. Okay, so here we want to use a simple way to overcome the problem. So we represent the molecule, use this called Morgan fingerprint. Okay, Morgan fingerprint is one type of fingerprint. You just count the number of the functional group around the molecule. So basically what it does is you take an atom, you ask what's the neighbor around the atoms, from a certain distance. And if you find special atom can be defined as a functional group, you just record this as a presence of one functional group. And then you increase the radius to search for bigger radius to go through more atoms. And you just keep going bigger circle and bigger circle. So intrinsically, you are correcting the short distance functional group and the long distance functional group in a 1D array. So that's what we do for the Morgan fingerprint, okay? So here we do the Morgan fingerprint for the solute, and we also do the Morgan fingerprint for the solvent, okay? Because the solvent is also a molecule, okay? So you can construct the solute and solvent with exactly the same procedure. So the systematic error hidden in this approach should be the same for both solute and the solvent, okay? And also for doing this, you don't need to know three-dimensional structure because you just count the fingerprint, okay? So even you don't have the structure, it's okay, you can do this. Okay, now you collect the fingerprint and then put everything together and to represent one measurement. One solute with one solvent, one Morgan fingerprint for solute, and one Morgan fingerprint for solvent, okay? Now let's clustering it to see how it goes, okay? For this one, this is just unsupervised clustering. We want to check how this fingerprint is distributed in the reduced dimension. This only has a solute fingerprint, no solvent, okay? We just pick the solute fingerprint. We use 1024 bits to represent the solute. And you can see it's okay because it's very dispersed, okay? Which means this fingerprint do fairly good to describe the difference between each molecule, okay? So this is a good representation of a good data set. But when you do the experimental measurement, if you combine the solute and solvent together as your input information, you are creating chaos. This is what it shows here. If you just add 1024 bits for the solute and 512 bits with the solute and add them together as your data set, and then you do clustering, you see the problem. They start getting together, gluing together, which means if you use 1024 bits plus 512 bits to represent a measurement, you cannot distinguish between the measurement because data points stay together. It's even worse if you add more data for the solvent, even worse because this is worse than this. So you see the problem. You want to add the solvent description. You cannot add too many solvent description. Otherwise, it screw up your data set distribution. Okay, so let's, can we solve this? That's what we learned from this analyze. Okay, so we want to do this two-stage. The idea is so simple, it's very simple. We just do two-stage. So the first stage, we do random forest with just solvent, solute, because we know the solute distribution is good. But doing random forest to predict the solute in respect to the experimental measurement should contain an error because it has solvation effect, right? So it should not be perfect, but that's okay. We do one stage random forest, predict the property, and we take that predicted property to the true experimental, that error. We do second stage random forest, including solvent, to make up that error. And then we add these two predictions together as our final prediction. That's it, two-stage random forest. Nothing new, okay? The first stage is to determine the solute contribution, and the second stage predicting the solvation correction, and then add them together. So at the end, you do this, that's the error bar. 11 nanometer to experimental value, including the solvation effect. So in this case, you don't need neural network. This is super accurate already. If you just cut it one stage and the second stage, okay? And we can also examine the chemical knowledge behind this. We do this permutation score. The permutation score meaning you artificially switch the feature value, and then if you artificially change the feature value, that should increase the error. And you can take the increment of this error to argue does this feature is important or not. If this is feature important, if you artificially change its value, that should give you huge error. Okay, that's called permutation. Okay, so here we examine the permutation importance of the solvation. That's less than 50%. Traditionally, if you do this problem with everything together, the fingerprint, solute, and solvent together, just do one random voice at once and hope the random voice to pick the right splitting method to do the good prediction. That's your permutation error. Truly depend on the solvent contribution. Okay, the number of the parentheses represent how much does this solvent contribute to this prediction. Okay, this is smaller. This is way bigger. Okay, and this gives you more chemically intuitive result because solvation should be minor than the solute contribution. Okay, so that's how we learn about this study. Okay, and I'm going to borrow 15 minutes to go to my final talk. And this is, we are doing the revision of this. This is related to analytical chemistry. Okay, and that's why it's used generative AI. This is not Shannon model anymore, but it has some generative model, and we think it's interesting to show people can we use generative method to do the application. And this is for Raman spectrum prediction. Okay, so the problem is the targeting a molecule is amino acid. Amino acid, we know there are like 20 plus amino acid, different type of structure, and then it relates to many biological process. So experimentally, if you take sample from a human or animals, you can do some dilution, and then you can measure the content of amino acid in that sample, and you can use that information to interpret it. Does this sample has some, disease or biological problem, things like that. So analytically, determining what is the dominant amino acid in the sample is critical. Okay, but amino acid from the structural point of view, they are very similar structurally. Only the side chain is different. Okay, so theoretically or experimentally, if you take the sample, that means all the signals are mixed with each other. Can you do a quick identification what would be the biggest contribution in this mixed sample? So that's the real problem. Okay, so in this case, we kind of cook the problem because we are approached by our colleague, analytical colleague, to solve their problem. So they are concerning to distinguish these two amino acids, the phenylalanine and the glutamic acid, and the transformation from phenylalanine to glutamic acid is so-called transamination effect. Okay, the transamination process, and this process involved in many diseases, including Alzheimer's disease. Okay, so if there is an analytical probe, can quickly identify does glutamic acid is more than the phenylalanine in the sample, that has huge biomedical effect to solve the problem. Okay, so we decide to cook a method to do this. Okay, so this is the Raman, and the Raman is very sensitive. The detection limit of the Raman is like a millimolar, even micromolar. So you just need a little bit, and you can see the Raman signal. So experimentally, people love Raman. Okay, but the Raman, when you have a complicated sample, the Raman signals stick together, and then it's very hard to read. Okay, so we want to combine this with DFT because DFT can calculate Raman. You have DFT package, you can compute the Raman signal of one particular molecule. So we compute the Raman spectrum theoretically with phenylalanine and glutamic acid. Okay, that's the Raman DFT spectrum. And then we train the random forest because we learned from the previous test, random forest is too powerful. So we want to stick with random forest. We just randomly take combination of these two spectrum together to generate a training set of mixed DFT spectrum of a binary amino acid from phenylalanine and the glutamic acid. And we can, because we construct the data set, we can pick any percentage we want, okay? Because we pick the percentage, so we know the answer, which one is the dominant one, right? We know the answer at the very beginning, okay? So we construct 20,000 arbitrary mixed DFT spectrum among 20 amino acids. So from 20 amino acids, we pick any two, any combination, and just do convolution of these two spectrum together, and then we form a new spectrum. And that's one of the training set, okay? So that's our training set, and then we train random forest. And we ask random forest which one in this mixed spectrum is the dominant species. The prediction rate is 99% if you train with DFT spectrum, which means the DFT spectrum is so clean. All the features precise. Even you stick any two amino acids together, random forest can still tell which one is the dominant one. Okay, so the good news is DFT spectrum is clean. The bad news is DFT spectrum is nothing to do with experimental spectrum. If you take DFT spectrum, you look at it experimentally, you cannot interpret it, right? I think Kenny knows this a lot. Yeah, that's the problem, okay? So this is just for fun as a computational guy, okay? So at least we know DFT spectrum is clean, and then random forest can tell which one is the bigger contribution from the DFT spectrum, especially out of 20 amino acids. So any two, you can do this. So the next stage is we do the same training with experimental spectrum. This is our experimental spectrum from my colleague, the real spectrum, okay? The way they purchase the sample and the pure sample of amino acid, and they make the sample on a substrate, and then they synthesize the amorphous solid film of that amino acid. It's under certain level of dry condition on a substrate, and then measure the Raman scattering of that spectrum. Okay, that's how they collect this. And then they do multiple times. Okay, but because it's amorphous film, which means every time they do the measurement, the molecular packing is different because it's an amorphous film. Okay, so they do multiple times. So we just borrow this experimental data from them, and we do exactly the same thing. We just make a random combination, okay? Any two. But here, because my colleague can only give me the glutamic acid and the phenylalanine spectrum, so we generate simpler training data set to 20,000 arbitrary ratio of two experimental glutamic acid and the phenylalanine data set. Again, we fit to random forest. Rate 0. Too complicated. Random forest cannot tell. You see the huge difference. This is experimental spectrum. If you do with experimental spectrum, random forest will fail. You cannot tell any of them. Okay. So now the situation is we want to transform the experimental spectrum style to the DFT spectrum style. That's it. These days, if you have a selfie, your photo, you can upload online and ask AI to transform your selfie to any style of feature, right? Exactly the same. That's exactly the same. That's variational autoencoder. So you can transform the spectrum from one style to another. You just need to train this transformation, okay? So this is called variational autoencoder method. The input here, this is experimental spectrum, okay? So I'm going to go to a little bit detail here. And this is our batch size. This is the wavelength scale of that spectrum. and we only concern 500 to 2500 wave number because that's the main signal region of the side chain. Okay, for the amino functional group and the alcohol group, those are two. We don't care. We just care about the side chain. Okay, so the experimental data coming in, and we form the latent space. And then after we form the latent space, we project to the DFT space, and then we ask the AI to compute the difference between the DFT signal and the experimental signal. And then we feed the error back this way. And then we keep training, okay? So here you can see here, by doing this, we need to do construction of two loss function. One is this reconstruction function, and this term usually just means the difference between DFT and the experiment at certain frequency region. Okay. The other one is Kullback-Leibler divergence. That's how we control the distribution of the latent space, and we sum this together. That's a very typical VAE approach to build this neural net. So after we form this, you can see here, these are two spectra. The orange one is the DFT spectrum. And if you take exactly the same combination from the experiment, after the variational autoencoder, that's how you get. So now you can see the spectrum, they align with each other very well after you do the VAE transformation. We just change the style. In more precise way, we kind of doing peak shifting, peak broadening, whatever, in order to make this peak to like DFT peak. That's it. Okay, but through a more systematic generative AI approach. Okay, I'm going to skip this. It's too technical. Okay, so after do this, we can summarize. Okay, if we just use random forest or another similar method called XGBoost, this is also a decision tree method, and you ask them to predict the experimental property, the accuracy is 0. It's too complicated to tell. Okay, so we need to denoise. Okay, the denoise involves two parts. Let me go back to the experimental. Okay, the first one is we need to make the peak sharper because experimental measurement, they are not as sharp as DFT. You look at the comparison of this. This is the experimental peak. This is DFT peak. DFT peak, the reason random forest can tell among the DFT signal is the peak is so sharp. There's so many information here. So it's very easy for random to tell what this is what. Okay, but experimentally, yes, very complicated photophysical effect. So peak become broadened. And the background noise, this is the background noise. We did not artificially shift this spectrum. This is exactly what they gave me. The background noise is huge because it's Raman scattering. There are huge background noise. So we need to remove the background noise and make the peak sharper to look like DFT. That's what variation autoencoder does. Okay. So if you can build a VAE like this, you can transform. Now, the prediction it becomes better and better. Okay. Here, we call this, what do we call this? Oh, deterministic limit of the VAE. What it means is when we construct the latent space, it has, usually when we construct latent space, we are looking for a Gaussian distribution. If you want to throw away this idea, just care about the final difference between DFT and the experimental, and you can call this deterministic version of VAE. Okay. So when we do this, we can have almost 100% of prediction as the original one. So means now my colleague give me any combination of amino acids. We fit into this transformation, and we can tell what's the dominant amino acid in this mixture. I think this is a huge application for the biomedical probe. Okay, and it just need random forest. You don't need neural net. Okay, of course you need some transformation anyway. Okay, yeah. So we analyze the latent space. Okay, so this is the experimental data when we project on the latent space. It clearly shows this have the blue dot here are the experimental measurement project on the reduced dimension that represent the dominant species glutamic acid, and the green or the orange one is the phenylalanine. And then there's a clear division from here. Okay, so that's why you can tell this is PHE or this is glutamic acid. Okay, and then if you do the conventional VAE to constrain the Gaussian distribution, that's what you get. A little bit messy, and that cannot give you a good reason. Okay, finally, this because we are doing the revision of this manuscript, we are asked by the reviewer, does the VAE really learn chemistry? We need to prove this. So that's how our argument is. Okay, so in the experiment, as chemists, we know if you want to tell this is a glutamic acid ramen, okay, we know this is the feature because it has sharp peak, right? Okay. This is not feature because it has no signal. So from chemistry, we teach students if you see a peak, that represents some functional group is doing something, right? So that's the important part. Students need to stick their eye on it. Okay, so this is the feature, this is not feature. Okay, so we take all the data on the reduced dimension and we plot one of the dimension. This is one of the dimension in respect to the ratio of this binary combination, because we are all the data, they are binary ratio. So this axis is the ratio of the glutamic acid. This is one of the dimension. And then if you plot together, that means this is linear. That means after learning, the model is already can project the weighting percentage of the combination on the reduced dimension. That's how this is linear. Okay, that's the first thing. Okay, so each data plot here is one binary data. And if you color this data set with the intensity of this region, 573 wave number, this is this peak area. You can see the linear distribution here has very clearly signal. When you have more glutamic percentage, you should expect higher intensity of 573. This is on reduced dimension. So it clearly teach you the AI model already identified 573 is something you should look at. And it gives you very good proportionality on the reduced dimension. So that's what this plot is teaching us, okay? And I can give you an opposite example. This is the same data plot, but if you color this data with the signal of this 1800 wave number, okay? So the distribution is exactly the same. I just color it with different data. The color here, I use the non-feature area that teach you this. There is no clear data color distribution. Okay, the color represent the intensity of 1800 wave number is irrelevant to the glutamic acid weighting. This is also chemistry because that's how we teach students. If you keep looking at this, you cannot tell what glutamic acid percentage or concentration, right? So with these two plots together, we want to convince the editor, our model actually learned the chemistry because it determines certain wave number region that's related to certain functional group contribution, and it has good proportionality, okay? So I'm going to draw the conclusion here, and then also directly predicting experimental properties, it could be done, but you need model tuning, feature engineering. But in order to do this, you have good domain knowledge. So that would be important to collaborate with experimental colleagues. You need their domain knowledge, and you use that domain knowledge to transform your design of the model. That's very critical, okay? And then we kind of promote low dimensional representation of the model. For any molecule, if you can represent that with low dimension information, that save you the preparation of the data set. And that also open a new dimension when you want to integrate all of this into large language model. Because large language model is text-based. These days, all the large language models, they are text-based. If you want to link your model, your small model with the large language model, you better use text. It's easier to build together, okay? And then the last one is spectrum transformation to the theory base. It can be a new idea. You don't have to tell the experimental always. Transform it to something else. Easier to tell, okay? As long as you can transform, okay? So that would be my conclusion today. And I would thank my student, also my collaborator, Berlin, which is our computational linguistics expert in the campus. He's my mentor. I learned everything from him, a professor in computer science. Okay, so that's how I learned this. And also my experimental colleague Yixing, who majored the Raman for us, and my student. And thank you for your attention. Yeah.
I have around 2 questions.
Sure,
in
sure.
2 parts.
Yeah.
So the first thing is to ask about your Shinet BS.
Yeah.
And Morgan fingerprint.
Yeah.
So I can see that if it is a Morgan fingerprint,
Yes.
it is is it also based on like the bond step?
Yes.
Looks like bond step
Yeah.
concept, right? So can you tell us like roughly like difference like for your one step model? So that so your featureization doesn't account for everything in the whole molecule, not just limited radius
okay.
or something like that.
I see. Okay, that's a good question. The Morgan fingerprint has to define the origin. So if you say I want to do 9 layer of counting, which means you determine one center atom, bound step 1 layer, bound step 2 second layer, and third layer, and then it can switch to another center atom, and you do the iteration through the whole molecule. So that's how it constructs the whole 1000 bits. So at the end, the artificial parameter here is how many layers you are looking for build this finger. You can go unlimited anyway. Yes.
And then the second thing is would be about the Raman.
Yes.
Raman Peak project. So for the transformation of the Raman Peak project, it like does the training set of your transformation come from the experimental setup with the same condition? Same environment?
Yes, good question too. Yes, let me show you. Yeah, okay. This is our experimental data. Okay, the green one is the glutamate acid, the red one is the phenylalanine, and this is a PCA analysis. So you can even experiment, they are kind of spread out. So there's a human effect. And also, as I mentioned, this is amorphous film. Every time student make it a little bit different. A student make different film from the B student. But at least they are still kind of, within certain range.
Okay, so you
Yeah.
mean that it's only trained based on the ensembles of Raman experiment.
Yeah, it depends on the training of the student. If you ask 30 students to do the same measurement, you see this. Yeah, but my point is, these 3 dots here, they are the DFT spectrum, the original DFT spectrum. So you look at the relative distribution of the DFT spectrum and experimental spectrum. They are different.
And that does that also mean that in real practice, and if I really want to bring a solvent and then to do a Raman
Yes.
Raman spectra and
Yes.
then to determine the ratio between different amino acids.
Yes.
So then it could be used anyway, right?
Yeah, in a Morgan fingerprint, you can say I'll take 1,000 bit for solute, maybe 200 bit for the solvent, and then stick together. Yeah, you can do this way.
Also, yeah, maybe later on, I would like to ask you about like the 2 steps random forest. So
Yeah.
So what was the output of the first step random forest? Does that a connection between like the second step of random forest?
The first output is the exact wavelength value, and that's the predicted of the first stage. And that after you get that value, that also can estimate the error to the true experiment. And so after you get the first stage value, you also know the error. And then the error is used for your second stage random forest. Yeah, so you estimate the solute contribution at the first stage, and then you estimate how much solvation error cancellation effect I need to add. And then you add this together as your final prediction. Yeah.
Thank you very much. So
Thank you.
is there any other questions?
Yes.
Okay.
We met before.
Yes.
Yeah. You were presenting your lab last time.
Oh, right, right. Nice meeting you again.
Yes.
And my question is when you train this Raman,
Yeah.
this data, do you need just the IR of the pure amino acid,
Yes.
or
Standard.
you need mixed
No,
methods?
I don't mean mix.
Oh, and then you mix them with different ratios.
We just convolute the 2 experimental data together. So there is an assumption in this convolution, meaning those 2 amino acids will not interact chemically.
Oh.
But typically it could be true.
Okay.
Yeah.
So that one is the DFT data that you mix, or the experimental data, or both?
Both.
Or both.
Yes.
And if for the real sample, it could be also some other amino acid and maybe sugar, carbohydrates. And would that also like will this model work with those actual sample?
It can be done following the same procedure. If you have saccharide or amino acid mixed together, you just prepare the pure sample of this, and then use the same combination to cook the training data. Yeah.
Okay.
We didn't try. Now we are testing because this example is just binary. We are testing 3 amino acids together. You can imagine when you have more complicated combination, it's getting tough. Yeah. So that's just.
I see. So this model right now is work based with 3 amino acids.
We are trying the three amino acids.
Okay.
Yes.
Okay. So I'm wondering, because if you want to diagnose the real patient, right? Maybe
Right.
take blood or some other urine or something like that. Is this applicable?
Not directly applicable at this stage, because we kind of we have another collaborator. They are doing the microfluid device. So the separation usually on that side.
Oh.
Yeah, so we kind of take the signal from very final stage, or just a little bit, much cleaner condition, and then predict. Yeah, so
Oh.
that's the design of the approach. Yeah.
So you recommend the separation before?
Yeah, you can have these days the microfluidic, they are super powerful. You just run the sample through certain stage. Just remove some. I think the key is you want to remove. At this problem, we are targeting the wavelength between 500 to 1800 region. We want to remove anything can contaminate this region. That's why we need a microfluidic. Yeah, so we kind of say, okay, because we want to tell the signal of sidechain. And this region is the most important experimental feature region. So from the domain knowledge, you probably know what could be contaminate the signal here. And then so we have to use microfluidic design to remove those.
I see.
Yeah.
So, like if this technique gonna be used, for example, with non-destructive
Yeah.
sample.
Raman
then-
is non-destructive.
Yeah. But if we need to separate, then we need to probably work with the sample, like to extract them, grind them, something like that. So, in your point of view, do you think this Raman with this simulation will it be applicable to use with the non-destructive sample? Like we don't need to purify it, things like that.
Because the algorithm here is not complicated. It probably can manage by a very small microchip. So, and microfluidic, I think these days should be able to integrate with microchip together. So probably can do just a small chip. It should be able to. My understand. I'm not a microfluidic expert, so should be done.
Like if we put the patient into a chamber and put the
Yeah, just put a drop of blood and then run through the microfluidic. At the end, and there's the microchip to operate this algorithm to tell what's the dominant composition. So that's just one major. Yeah, so it should be theoretically possible, but I have not done that yet. Yeah.
Okay. Thank you very much.
Yeah, thank you.
Okay, so thank you so much, everyone, again, and thank you so much, both Professor Brad and Kenny, for coming to KBCT, which is quite far away from the center of of Bangkok, and then you have to catch a car to go to Kola to go to Anse this afternoon.
Yeah,
So
Kola, yes.
yeah, it would be it is my great pleasure to have both of you here. So this is the end of the NNP school, and thank you so much, everyone, and thank you so much for those who are watching this in the YouTube live and or recorded video later. So without further ado, please give a round of applause to the two speakers today. Thank you.
Thank you.
And that's it, guys. Let's go have lunch. Okay. Yeah. Let's take a picture together. I think we have a new.
Are you living with me together? Or?
Yeah.
Okay.
Oh, you're working with Thana?
Mode of transport.
Where are you from? You're not Thai.
Grab? Is Grab okay? with
Morocco.
you?
Oh, okay.
No, actually,
good,
I'm
good,
okay
good.
with anything. So
And you are working.
Grab is easier.
Yeah.
Like
The fattest one.
Right.
internship.
Unless you
student
want
or
to
something?
spend like 4 or 5 hours.
Oh, for how long? 2 months.
Oh,
good.
sorry.
Okay, let's take a picture ตรงโซฟาเลยครับเอาที่มีป้ายได้ไหมป้ายหรือข้างหน้างั้นผมกดN ไลน์แล้วนะ
Yeah, it's that the one I put the other one. Yeah.